go ahead and get started. Couple of quick announcements. Again, those of you who have been looking at the syllabus I mentioned last time, on February 9th, there'll be a guest lecture. Dr. Bartlett's, I think it's March 9th, we're also going to have a guest lecturer. One of my graduate students, George Kaysen, will be talking about CRISPRs and small RNAs. It's also listed in italics in the syllabus. Other questions? Someone, I think it was Damon, I forget your last name. Thank you for sending me an email first thing this morning. I posted the keynote version of the lecture notes last night and didn't do the PowerPoint ones, which I did again this morning. So if you notice something like that, you're like, oh, please send me an email, and I will try and get on it. Thank you, Damon, wherever you are. Third thing is clickers. I know a number of people have sent me emails about how to register their clickers. They're having trouble doing it. They can't find the course online, etc. To be perfectly honest, I don't know how all of that works. They keep changing the system on me. But clearly a lot of you have figured out how it does work. So please just post, if you have registered your clickers specifically for this class, go on to the discussion list and post how you did it. And hopefully then the other people who have questions about that will be able to figure it out. So then any questions about sort of, syllabus, clickers, etc. All the clicker scores are coming in. I'm getting them. If you have a score in D2L for clicker that's anything, then you're good. You're registered. It's working perfectly fine. Other questions, comments, worries, etc. No class on Monday. So do your Martin Luther King Day volunteering things, etc. You can come here if you like. I won't be here. So, yeah. Is your clicker score under the session total? Yeah, so the question is, where do you see the clicker scores? They're just under sessions right now. So session 1, session 2. Session 1 was lecture 1. Session 2 is lecture 1. Those numbers are not going to count. Clickers are actually going to start counting as of next Wednesday in terms of calculating scores, grades, etc. Other questions, comments, concerns on this? Okay, so today I wanted to finish up on our discussion of all the chemistry you need to know for molecular biology, which again is a ridiculous oversimplification, and then talk about proteins a little bit. We'll talk much more about proteins when we talk about translation a little bit later on. But to start with here, is this bright enough in the back, or need to turn the lights down? Works? Okay, yes, no? Lights down a little bit? Okay. Let's see if I can... That's down a lot. I'm not sure how much more I can change that. That's kind of dark, but will that work? Okay. No falling asleep? I won't notice anyway. So to start with, again, the chemistry here, this is partly because the textbook, to hopefully save you a tiny little bit of money, although it's ridiculously expensive, is that the same textbook is being used for cell biology and for molecular biology. So a lot of this stuff is much more of a cell biology approach, but a couple of things here are really pretty important. We've already talked quite a bit about water being absolutely essential for biology as we know it. So it's really all about different water reactions and breaking of covalent bonds that have to do with carbon. And so basically that's all the chemistry we're going to talk about in this class. They're going to be condensation reactions, hydrolysis reactions, and those are going to be involved for 90% of the time in breaking and forming carbon bonds. So hopefully, again, this is all review. If you think about photosynthesis, you need to get energy into life systems somehow. For the most part, that comes through sunlight. Yeah, there is some sunlight even with the crazy weather we have here, which will then allow the incorporation of carbon from CO2 into basically food, which we use, and then convert that food back to CO2. We take up oxygen and create water. What kind of reaction is that going to be? It's a condensation reaction because you're releasing water. And then just cycling, this is looking at carbon cycling on Earth. Basically, it's all about the CO2, mostly in the atmosphere, dissolved in the water. Photosynthesis allows it to be picked up into plants, algae, and bacteria, which can then be eaten by animals, which produce CO2, and so on and so forth. Burning fossil fuels, of course, changes a lot of things about CO2, and we won't get into that as far as this class is concerned. So all these reactions, again, wonderful reaction, let's say creating hydrolysis reactions, it's great, it's wonderful, you increase disorder in the universe. Why don't all these things happen spontaneously? Why don't we all combust immediately? That has to do with activation energy, and of course, hopefully, again, this should be review for you. The activation energy is what sort of allows any reaction to not take place. You have to have a certain amount of energy added to even a so-called spontaneous reaction or an energetically favorable reaction, which is just shown here. Total energy of X is lower than the total energy of Y, but Y doesn't spontaneously form X because it has to get across this so-called activation energy barrier, so you actually have to add energy to Y before it can become X. And we're going to talk about, to some extent, enzymes. If you're really interested in enzymes, take biochemistry. I'll talk a lot more about enzymes. But enzymes are basically just catalysts. Catalysis, all the catalysis does is lower this energy barrier. And why is lowering the energy barrier so important? The reason for that is if we didn't lower energy barriers, basically nothing would happen. And particularly if we're talking about the vast majority of reactions that we'll talk about in molecular biology, which are polymerization reactions, going from monomers to multimers. And monomers to multimers, as we talked about last time, is energetically really bad. It's really hard to do that. And so if you didn't have all of these enzymes to help out and stimulate some of these reactions, none of it would actually happen. So the other thing that bears in mind, even with catalysis and even with that lowering of energy barriers, allowing things to happen, most of the reactions, again, I'll just take polymerization as the example, are energetically unfavorable. Now, so what do I mean by energetically unfavorable? Energetically unfavorable means you have a positive delta G. If delta G doesn't mean anything to you, you can either read this part of the textbook, actually a nice chemistry review here, or think about maybe doing Gen Chem and coming back and doing this course some other time. But the idea, if you have a positive delta G, i.e. you're going from a low energy state to a high energy state, this is never going to happen unless you have a coupled reaction, an energetically favorable reaction, which has a negative delta G. And this is just diagrammed here. Basically, you have your first reaction, which has a negative delta G, i.e. it's going to be an energetically favorable reaction, a spontaneous reaction. You may need to catalyze it as well. But that then has to be coupled to a reaction which is otherwise energetically unfavorable. And a lot of this coupling, the little wheel that we have right here in the middle, that's due also to these enzymes that are bringing these two different reactions together. And last time, I think I did a bad job of explaining the whole idea of the fact that most of the reactions we care about in molecular biology are energetically non-favorable, but pathways are energetically favorable. So basically, you can think of this here as being a pathway because you've got two different reactions. What you want to do is get X to go to Y, but to do that, you have to have C go to D in terms of these actual reactions. And we talked a little bit about that. We talked about the different energetics of reactions again last time. So that's the overall picture again. If you want to refresh your memory about delta G's and energetics, again, recommend going back and looking at that section of the book. Yes?
So the reason why it has to be coupled is so that the energy can go somewhere, correct?
Yeah, so the question is, why does it have to be coupled? And the answer to that is, if it's not a coupled reaction, that energy which is generated from that first reaction is dissipated as heat. So it just increases disorder, entropy that we talked about last time. That was what happens to my kids' rooms if I don't force them to clean them up. So, yeah.
So the reaction with the negative delta G always has to have a greater delta G value than the positive one, correct? To allow the other one to, or is it situational?
Yeah, so the question here, I'm just going to repeat it, so it ends up here in the lecture recordings. Those are working, by the way, lecture recordings? Yes? Working? People looked at it? Good. So the absolute value of delta G has to be larger in the one which has a negative delta G than the one which has a positive delta G, because that's the first law of thermodynamics. Energy is not being created. Other questions on this stuff? So yeah, this is great. It's wonderful, yes, but how do you do this coupling? How do you go from one reaction with large negative delta G to something which has a smaller positive delta G? And for the vast majority of biochemical reactions, and the ones that we'll be talking about here again in molecular biology, there's this concept of what's called an activated intermediate. And so 90+% of the time here you start with ATP, which is your high energy source. This has been generated due to the breakdown of your fixed carbon, usually due to photosynthesis. You generate lots of ATP. That ATP then gets used to generate a, again, high energy intermediate. And the main thing here is that we have now a bond which can be hydrolyzed and then exchanged to give you what you're actually looking for. So going back to, I should have put up the image here with the two different arrows on it. So that big negative delta G arrow is going from glutamic acid to this intermediate, and the smaller positive delta G is going to glutamine. You can't go from here to here energetically without having this extra step and basically using the energy of ATP. And this is what we'll see again and again anytime there's a reaction we're talking about polymerization of nucleic acids, translation, etc. All of this is going to be based on this high energy intermediate. And all of these reactions are catalyzed by enzymes. So what about polymerization? Same thing is true actually for polysaccharides. We're not going to talk about those in this class. But here for proteins, and we'll talk a lot more about proteins as we move to the end of today's lecture, you have a short protein. This can be as much as 1 or 2 amino acids that has an extra monomer that gets added to it. All of that is a condensation reaction, and that's driven by ATP hydrolysis. Polymerization of nucleotides, this is actually that exception where you're not always using ATP. Sometimes you're using other nucleotide triphosphates in this particular case. But again, it's a high energy intermediate that allows you to go from a shorter molecule to a larger molecule. Let's look at nucleic acid polymerization right now, just at a chemical point of view. We'll talk a lot about the enzymes in a couple of weeks. Here, one nucleoside just has 1 phosphate on it. A nucleotide is going to have 3. Monophosphate. Here, 2 ATPs are used to add 2 phosphates here on your high energy intermediate. This then will combine with an already extant, either 1, 2, or for that matter, many nucleotides. There's an OH here, which is going to get attached here to the OH, which is connected to these phosphates. That 1 phosphate is going to be left. These 2 high energy phosphates, that hydrolysis again is giving us the impetus to get our excuse me, reaction here. This is still positive delta G. Anytime you're going from a monomer to a polymer, that's going to be reducing entropy. It's like cleaning up the room. That requires energy. So that energy, where's it coming from? It's coming from this activated intermediate and the ATP that we had in the first place. In the case of nucleic acid polymerization, it's not just the hydrolysis of this bond right here between the first phosphate and the second and third phosphate. There's another reaction, another hydrolysis reaction here, which cuts off this last phosphate as well. So you just end up with two phosphates here. And because of this, first cutting off what's called pyrophosphate, the two phosphates together, and then going to the individual phosphate, this makes this reaction, this polymerization reaction, basically irreversible, because putting those two phosphates back together is not going to happen. And there are a few enzymes that will do that, but they're very few and far between, and it's extremely high energy reaction to bring those two back together. So that's all of chemistry that you need to know for molecular biology. Isn't that wonderful? Yes? No? So just as a review, last time we talked about the number of atoms that we need. It's basically carbon, hydrogen, nitrogen, oxygen, a little bit of phosphorus and sulfate, but the rest of it we can kind of ignore. Great, rest of the periodic table. Weak interactions, and we're going to talk much more about these weak interactions as we move along. The covalent bonds, that's actually a lot of the stuff we're talking about right now. To get a covalent bond, you have to have all of these coupled reactions. Most of the interactions we'll be talking about, other than these polymerization reactions, are going to be much weaker interactions, and lots of weak interactions. Those are going to be either van der Waals interactions, hydrogen bonding interactions, and ionic interactions. We'll talk about some of those ionic interactions more with the proteins as we move along here. Everything we know about life is completely dependent on water, and one of the main reasons for that is water is a polar molecule in the chemical sense. It's got a slight positive charge at one side, a slight negative charge at the other side, and it loves to form hydrogen bonds with itself. And that keeps water liquid at relatively high temperatures, but it also is really important for interactions with all kinds of other biological molecules. The strong interactions, again, it's mostly what we talked about today so far. These are going to be the covalent bonds which form, and in order to get covalent bonds, particularly in terms of polymerization reactions, you really need to have activation energy. That activation energy has to be lowered. How do you lower activation energy? With catalysts. What are the vast majority of biochemical catalysts? Are enzymes, exactly, which are almost all proteins. To get, again, polymerization to take place, we usually need to go through these high-energy bond intermediates. That means you have two coupled reactions, the one which has a large negative delta G and one which is a slightly smaller positive delta G. And then this precursor activation has to do with the high-energy bonds here. For those of you who don't feel completely up to speed on the chemistry or maybe a little confused about some of the stuff we talked about, other than me just not talking about it very well, the part of the textbook which I didn't bring with me, there's some blue pages in the textbook which are really nice review in Chapter 2 of the chemistry which we've just talked about here, particularly panels 2.1 to 2.7 in the textbook. So it's really quite a nice model there. So any more questions on chemistry before I ask you a question? No? Okay, so let's do this question thing. Almost all polymerization reactions in biology are blank reactions, acid-base, condensation, hydrolysis, dehydrogenation, or reduction. And again, feel free to talk to your neighbors about this. This is the part where everybody should be getting 100%. 10, 5, 4. We're not 100%. The basic question seems to be between condensation and hydrolysis. Basically, the real question here is what's polymerization? Yeah. Yeah. So, well, that's a great answer. I'll get back to that in just a second here. So, condensation and hydrolysis. What are we talking about there? We're talking about water reactions, right? So, condensation is doing what? It's taking water out. Hydrolysis is putting water in. What's present at the highest concentration around? Water. Water is present at way higher concentration than anything else. Again, we looked at that last time. What's the concentration of what the things in cells? It's mostly water. So water is at very high concentrations. If you're increasing the amount of water, is that going to be energetically favorable or unfavorable? Unfavorable. So if that's energetically unfavorable, what is polymerization like? Going from monomers to multimers, arranging it, getting more information, is that favorable or unfavorable? Unfavorable. So what do we end up with? They're all going to be condensation reactions. So hydrolysis is breaking things down. It's breaking those bonds. Polymerization is making bonds. And so those are all going to be condensation reactions. Okay, so now I'm going to try something a little different. We're going to try coming downstairs and see how that works, because I hate being up here. May or may not work. We'll see. I can see a lot of you in the back here, too. So, and it allows me to walk around and get my nervous energy out. So for the rest of today, we're going to talk about proteins. And again, this is a whole chapter in the textbook. We're just really skimming over it. Yes, we will get to more details in nucleic acids in Chapter 4 next time. So start out talking a little bit about amino acids. These are those individual monomers which then get put together in terms of proteins. And we'll talk a little bit about how that happens. How they get put together are peptide bonds, and we'll look a little bit at how those then fit together as well. One of the really neat things about peptide bonds is that they're incredibly flexible. And so they can turn around in lots of different ways, which allows the proteins to have huge amounts of different structures, and lots of different structures means lots of different functions. Exactly. So what are those structures? Secondary structures, particularly alpha helices and beta pleated sheets. The vast majority of proteins are made up of these mix and matched parts of either alpha helices or beta pleated sheets. Those then come together, these secondary structural elements, to give you a tertiary structure. That's the overall three-dimensional structure of your protein. Many proteins form multimers, which is many individual polypeptide chain. And when I say an individual polypeptide chain, it has an N-terminus and a C-terminus to it. And we'll talk about N and C termini in just a second here. Any protein, and usually people talk about these as a protein complex that has multiple N and C termini, is going to be a multimer. These things coming together. And the vast majority of proteins that are doing things in our cells are multimers, going from dimers to much, much, much larger structures. And we'll talk about a couple of those as we move along through here. I skipped over domains / motifs. This seems to be a difficult concept, probably because I don't explain it very well. A domain, and I'll try and do this multiple times, is a part of a protein which has its own structure, and of course its own structure is usually going to have its own function. That's actually going to have a specific structure in and of itself. A motif is a sequence in your protein. Usually we'll talk about that as being primary sequence or primary structure. It's just a particular amino acid at a particular position. That then gives you some indication what the function might be. But that's not going to have any kind of function in the absence of the actual structure. So that's the difference between domains and motif. A motif is a sequence, it's primary structure, whereas a domain is tertiary structure. Then we'll talk a little bit about protein families and some modeling, and my remote here is not working. Let's see if this one will work. Yay, okay, good, it does. So proteins. Proteins, you can also just think of these as polymeric amino acids. Amino acids, I'm going to actually have to go back and up here at the top. Sorry about this. My phone's not talking to my computer, so I can't do the pointer as well. Amino acids, just like proteins, are made up of a particular structure which has a beginning and an end. So this is our molecular biology polarity that we talked about when we talked about the individual nucleotides a little bit earlier on. It's got an amino group at one end, that's your N-terminus, a carboxyl group at the other end, that's the C-terminus. So beginning N, end C, and then amino acids all have these so-called side chains that are hanging off on the side, and that gives you the individual amino acid that comes together. There are 20 normally coded amino acids. There are actually up to 23 different amino acids that the ribosome puts in, and we'll talk about those a little bit later. But as far as we're concerned for now anyway, just 20 different amino acids, and the difference is all in these side chains that are coming off of this amino acid. The chemistry that happens here, again, it's going to be a condensation reaction. It's creating water. It's when you have a reaction between the C-terminus of one of these amino acids and the N-terminus, the amino group, so carboxylic acid group, and the amino group here forming a peptide bond. This is just a single carbon-nitrogen bond that can rotate around, which gives you a lot of flexibility. The actual protein itself, this polypeptide, has huge amounts of flexibility because of this individual peptide bond. Because forming peptide bonds is always going to be between carboxyl groups and amino groups, that means that when these two have hooked together, now it's got an amino group at one end and a carboxyl group at the other. And so on and so forth. Your protein has an amino group and a carboxyl group, the N-terminus and the C-terminus. Each individual amino acid has this same structure as well. What kinds of amino acids do we have? Do we need to know the structures of all the amino acids for the exam? No. Would I expect you to know that there are different flavors, different kinds of amino acids? Yes. And a couple of particular ones that we'll look at on the next slide here. The important here, anyway, is to think about the nonpolar and polar amino acids. And this is now the chemical polarity. And I'm sorry about this whole polarity, molecular biology, N-terminus, C-terminus, 5', 3'. Chemical is going to be polarity interacting with water, as we talked about last time, slightly charged. So polar amino acids can interact with water. Nonpolar interactions are going to get squished together because water likes to interact with itself. So we're not very interested in most of these amino acids, with a couple of exceptions that we'll talk about in a second. We're much more interested in these because they're going to be in the outside of the protein, and there they're going to be interacting with everything else, like reactions which they're going to be catalyzing. If it happens to be an enzyme, it'll be interacting with nucleic acid if it's a nucleic acid binding protein. So these are going to be the amino acids that we're going to be particularly interested in, and most interested actually in these 4 amino acids up here: aspartic acid, glutamic acid, arginine, and lysine. Again, I don't expect you to remember the single letter code, but I find it to be very useful. D and E are aspartic acid and glutamic acid. R is arginine. K is lysine. Now, why am I concentrating so much on these 4 particular amino acids? Because we'll see them again and again and again and again as we move through the rest of the course. And why is that? That's because these amino acid side chains get ionized. in regular aqueous conditions, and aqueous conditions, of course, is what the cell is. It's mostly made out of water. So what these are, and we talked a little bit about basic versus acidic last time in lecture. Acids lose hydrogen ions, bases lose hydroxyl or take up hydrogen. So because of that, they end up with charged side chains. And these are you can argue about histidine, but basically glutamate and aspartate, or glutamic acid and aspartic acid, depending on if they're charged or not, will have stable charges to them. So glutamic acid, aspartic acid are going to have stable negative charges. Lysine and arginine are going to have stable positive charges most of the time in most proteins. What do positive and negative charges like to do? Interact with each other. Exactly. And so that's why these are those important ones. And the actual structures of the side chains are not important, but that these can be charged and usually are charged is the important aspect here. So we'll see where we need those in just a second. We talked a little bit about how interactions happen. Again, ionic interactions, the main ionic interactions we're going to talk about here are going to be those kinds of ionic interactions between these charged amino acid side chains and either nucleic acid or other charged side chains that are interacting there. So I mentioned already, repeat it again, different kinds of levels of protein structure: primary, secondary, tertiary, quaternary. Primary is just the sequence. So from N-terminus to C-terminus, that's going to be your primary structure. Just amino acid 1, amino acid 2, amino acid 3, amino acid 4, etc. Primary structure, primary sequence. Secondary is how each of these amino acids associate with each other based on those what we call backbone atoms. Now, what is a backbone atom? Backbone atom is everything except for that side chain which is different between the different individual amino acids. So if you have interactions between backbone atoms, that's going to be your secondary structure. Secondary structures come together to make tertiary structures, and it's this process, secondary going to tertiary, which is, for the most part, not dependent on those backbone atoms. It's going to be all those side chains that are sticking out. So the individual different amino acids are going to determine what gives you this tertiary structure, and this tertiary structure, in some cases, and actually quite a few of them, can come together either with more of exactly the same. In this particular case, you have 4 of the same, a quaternary structure. You could also have 2 of the same. You could have 2 different ones, etc. We'll see more about that. So the question that we're talking about here, and illustrated down at the bottom, is how you go from secondary to this tertiary structure. And again, it's all about these side chains, the different colors here representing the different chemistries of each of those individual side chains. So here, the bottom, green, nonpolar side chains, blue and red, polar side chains. Nonpolar means what? Pardon? Hydrophobic, not wanting to interact with water. It's all in an aqueous environment. Where are those going to be? In the inside of your protein. So you'll notice all the green bits are down here in the middle of the protein. My pointer would work. Here we go. Well, green down the middle of the protein, whereas all those colored ones are present on the outside. There are lots of different, sorry. Yeah.
The polar side chains are on the outside.
Mm-hmm.
Then the non-polar regions, that's what causes the polarity, or like the attraction.
Okay, I'm not sure I quite understand the question, but I'll go over this again. So what I mean about polar and non-polar side chains, if there are polar side chains on the outside, basically what happens? So those polar side chains the polar side chains on the outside will interact with water. And so they'll be perfectly happy there. If you have nonpolar side chains on the outside, those don't really want to be there because the water wants to interact with water rather than interacting with these individual pieces. And so what happens there is either those nonpolar side chains will interact with other nonpolar side chains in other parts of your protein. And so, for instance, if you've got a quaternary structure that looks like this, hang on, let's go back. A quaternary structure that looks like this, and we've got our all kinds of interesting things going on here. Quaternary structure looks like this. Maybe there's a polar side chain that's sticking out here. You know, it's going to interact with a polar side chain that's sticking out over here. So it's going to be interacting with some other kind of polar molecule. And we're not going to talk in this class very much about membrane proteins. Membranes are lipids. Those are incredibly polar molecules, nonpolar molecules, et cetera. So often if you've got, say, a nonpolar side chain which is sticking out, that's going to be in a membrane. It's going to be interacting with something else which is nonpolar. So in general, you're going to have your polar side chains on the outside, the nonpolar ones on the in one. Yeah?
So I think what I was trying to, what she was saying is, because you have the polar on the outside and the nonpolar on the inside, is that what causes the folding of the tertiary structure?
So what causes, we'll get to protein folding in just a second here. It's how you go from secondary structure to tertiary structure. Yeah, we'll get there in just a second. If that was your question, yeah, we'll be there in just a second. Okay, my phone is not talking to my computer today. I think it's actually had to do with, people having trouble with wireless today? Well, now it seems to be working okay. Yeah, I don't know what's going on with this, but be that as it may, say technology. So secondary structures, I mentioned before, secondary structures are really mostly about the interactions between the backbone atoms with each other. And there are two main forms of secondary structures, the alpha helix and the beta pleated sheet. Both of those actually were hypothesized by Linus Pauling, double Nobel laureate from Portland, Oregon. Went to that other school down the valley, but you know, details. So he and some of his students came up with these really brilliant ideas about how proteins could be being put together. And basically by building models, not unlike the DNA model that we have down here, and seeing how they would interact with each other. And the whole concept of the hydrogen bond really comes from the really amazing insights of Linus Pauling. I was fortunate enough to hear one of his talks soon before he died, and he was telling us all about how wonderful vitamin C was and how it protected against everything, including the common cold, while he was coughing the whole time. So yes, absolutely brilliant in terms of alpha helices and beta pleated sheets, but some of us aren't always right all the time. So, but what's happening with the alpha helix? What happens with the alpha helix is you have these individual hydrogen bonds which form between the, again, backbone atoms. So every single polypeptide chain has these. So you have an oxygen connected to one of the carbons in your chain, which is then forming hydrogen bonds with the nitrogen, which is on the amino acid, which is 4 amino acids away. And so because of this, you end up with a wonderful helix, and we'll talk a little bit more about helices as we move along. But probably the most important thing about this alpha helix, what does it do with the side chains? It sticks them out, perpendicular to the helix. So now all these side chains can interact with each other. If they're polar side chains, probably going to be going down to the middle of the protein. If they're nonpolar side chains, then interacting with something in the outside world. In many cases, for the rest of this class, we'll be talking about DNA. So the alpha helix is pushing all those side chains out perpendicular then to your helix. These helices are all going to be right-handed helices, and we'll talk about right-handed helices and left-handed helices a little bit more later on. I have a whole collection of helices up here at the front, so afterwards you want to come take a look at them. You may. I used to give people extra credit for bringing me left-handed helices, but I've got a whole collection of them, so you don't do that anymore. The other way that backbone hydrogen bonds are forming with each other is in the beta-pleated sheet. The beta-pleated sheet, again, has hydrogen bonds which are forming between oxygens that are part of the backbone and NHs, which are part of the backbone. And again, this is just because these are peptide bonds. But now instead of 4 amino acids apart, these can be very long distances apart. In fact, you have no idea how far apart these can be. They can be literally hundreds of amino acids apart relative to each other. But they do form hydrogen bonds with each other, forms also a very nice, more or less planar structure. What does this do with side chains? Sticks them out. Now, up and down rather than perpendicular to your helix, but again, having those side chains in such a way that they can interact with other side chains, they can interact with other molecules. Yeah?
So if we were looking at like a big protein, right, like you had earlier where it had helices and sheets on it,
Mm-hmm.
would they, like, these 3 right here count as 1 beta sheet, or would that be 3 beta sheets next to each other?
Okay, so the question basically has to do with definition, correct me if I'm wrong, of what do you call a beta sheet, what you call an alpha helix.
Yeah,
So,
like an alpha helix is just 1.
right, it's going to be 1 helix. But what do you call a beta sheet? So each of these individuals you would call a beta strand. When you've got more than one of them, and so you can have 2 strands that make a beta sheet, you can have 4 strands that make a beta sheet. We'll talk about amyloids, which are lots and lots and lots of beta strands that are forming not quite a beta sheet, but a very similar kind of thing. So you can have multiple. Each of those strands, as long as it's oriented in the same way, these can be either parallel or anti-parallel. And anti-parallel and parallel has to do with N and C termini. So you can have one of the strands that's N to C, the other one which is C to N, or vice versa. They can actually also be parallel to each other. The hydrogen bond, no difference one way or the other. But more than one strand will give you a sheet. Now, I wouldn't necessarily call usually more than one strand a sheet, but so be it. That's how we have these here. So any questions about alpha helices, beta sheets, secondary structures? And whenever Stedman says any more questions, what does he mean? He's about to ask a clicker question, exactly. So let's actually get this to move forward, if it will. Yes, no? We're not talking to ourselves here. Computers, we're having some serious technical issues here today. Where are we? Try one more here. There we go, yay, finally, clicker question. Protein secondary structures are due to van der Waals interactions between side chains, hydrogen bonds between side chains, van der Waals interactions between backbone atoms, hydrogen bonds between backbone atoms, hydrophobic interactions. Five. So one of my goals for this class is to get 100% answers, and it's actually really to get 100% right, not 100% wrong. So yes, it is hydrogen bonds between backbone atoms, which are there. So, and hopefully, backbone versus side chains. And if people do have questions about this, feel free also to email me about it afterwards as they're confused in some way. Just let me know, and I'll also discuss those on D2L. So now we finally get to folding. And maybe this is your question again, and thanks again in the back about this. So how do you go from secondary structures, which are hydrogen bonds between backbone atoms, to these tertiary structures, which is really what you need in terms of actually getting some kind of activity? It's all about the interactions between the side chains. We already talked about these acidic side chains and basic side chains interacting with each other. That's one way that they interact. You also are going to have your nonpolar amino acid side chains interacting with each other. And then, I'm not going to talk too much about it, but also cysteines. You can actually also have covalent bonds that form that give you your structure. So it's a whole combination of all of these different things. Hydrogen bonds, which are not shown on here. Oh, actually, there's up on this corner. We've got hydrogen bonds. So hydrogen bonds, van der Waals interactions, ionic interactions between the side chains are going to give you that structure. So that's the protein folding question. And how do you do this? Now, in theory, we should be able to figure out exactly how this works because we know all these interactions with each other. But unfortunately, because there are so many possibilities, in terms, I mentioned the peptide bonds, they can rotate in all kinds of different directions. We're still trying to figure out how you can go from primary sequence, just amino acid sequence, which actually is really easy to get because we can do DNA sequencing and we know what the genetic code is, to actually predicting a structure. Now, why is predicting a structure so important? Because we can then predict functions, hopefully, with this whole thing. And so that's the basic idea. Again, we're not there yet. I want to spend a couple of minutes talking about helices. Why are helices so wonderful? Because if you've got all these side chains, you stick them out on the outside. But just from a geometric point of view, helices are great because they're basically individual pieces that are getting put together in exactly the same way with other individual pieces, which is a quick shout out for what I'll be doing tomorrow afternoon. Just on the other side of the river over at OMSI, meet a scientist and build a virus is what my outreach activity is going to be. So if you've got something to do between, nothing to do between 1 and 4 tomorrow afternoon, come on over to OMSI and we can talk about virus structures and helices. But it turns out that viruses also use these helical structures in terms of putting things together. And again, it's because each of these individual subunits can fit together with the next one in exactly the same way and packages really nicely relative to each other. We do have both left and right-handed helices. I don't know if you've talked about this in terms of stereochemistry. Right-handed helix, left-handed helix. One way to think about it, if you have make a thumbs up with one hand. If you look the direction that your fingers are going towards your thumb, in this case it would be a left-handed helix. In this way it would be a right-handed helix. You can also, if you look down the helix, if it's going in a clockwise direction, it's right-handed. If you're going in an anti-clockwise direction, it's left-handed. And again, I've got lots of examples of helices up here that you can come and take a look at. And I think it's useful to take a look at these. And it used to be you always had phone cords. What's a phone cord? Anybody have a phone cord? Those are really nice. And I'll show you there's also some supercoiling things that we can look at a little bit later on with these things. But helices are a really good geometric way of packaging things together. So if you've got monomers of something, which is all the same, like the peptide backbone, or as we'll see for DNA, the phosphodiester backbone, packaging them together in a helix is a really efficient and useful way, just geometrically in terms of putting things together. One of the most famous of these helices interacting with each other is something called the coiled coil. This was found, I'm trying to think, probably about 20 years ago when people were starting to get high-resolution structures of proteins. What they found is lots of proteins actually have very large amounts of alpha helices in them. Again, not surprisingly, it's a nice geometric way of compacting things together, and has side chains sticking out on either side. But you were asking about polar side chains, and what happens if you have your polar side chains sticking out on either side? They could be interacting together in the inside of a protein, but you could also have them interacting with another protein. And so that's what people found with these coiled coils, also known as leucine zippers. Leucine is a classic, large, nonpolar amino acid side chain. If you think about how often the amino acids repeat, how many amino acids is it if you have your hydrogen bonds forming an alpha helix? It's 4 amino acids, and it turns out that where the side chain is every 3.5. So if you've got every 7 amino acids, you have a large hydrophobic or nonpolar amino acid that loves to interact with something else that has a large nonpolar amino acid side chain on it. And particularly that's leucine, you end up with a leucine zipper. So multiple leucines, 7 amino acids apart, going to be interacting with each other. So if you're just looking at a amino acid sequence, every 7 amino acids, it has a leucine. You can predict that it's going to be interacting with something and have this alpha helical structure. This is a prediction that you've just seen from the primary structure, the primary amino acid sequence. Is that a motif or a domain? That's a motif because you're just looking at the sequence. You make a prediction in terms of what kind of structure that would have. So these leucine zippers are really motifs. Turns out that leucine zippers can happen as 2 alpha helices that come together, 3 alpha helices, a little bit different. Collagen, which actually makes up a lot of our connective tissue, is also helices that are wrapped around each other. Not quite in exactly the same way, but a similar kind of process. Yeah. Figure out what's going on with these things. So domains, on the other hand, as I mentioned, are pieces of proteins that have their own 3-dimensional structure, and again, usually going to have a particular function. So here's an example up the top. They love to use SARC as an example in the textbook. That's partly because some of the authors actually work on SARC in their labs, so that's why SARC, you see SARC again and again in this textbook. But here, conveniently, the different colors are noticed. Here's a domain up at the top here in green. Here's another domain in yellow, one in orange, one in blue, etc. Each of those domains usually have, again, a separate structure and a separate function. And one of the things that we found in sequencing the human genome and looking at lots of different proteins is that often these domains get mixed and matched. And so that's what's shown down here at the bottom. Here you'll have different domains. So here's an EGF. Not important what EGF here is. So just an EGF domain, which is part of one protein. It only has one domain. But that same domain can be used in multiple different places, in multiple different proteins that actually overall have a different function. EGF here, GF stands for growth factor. So just a single domain is a growth factor. Factor 9 is important for blood clotting. So mixing and matching domains can give you different parts. So one part of this could be interacting with a particular small molecule. And that small molecule may be important for blood clotting. It may be important for stimulating growth. And so mixing and matching domains seems to be a way that through evolution, and we'll talk about to some extent how this happens as a recombinational technique that happens in the development of genomes, that mixing and matching domains allows you, again, biology, to give lots of different kinds of overall functions of these. People say there are relatively few domains, like a couple of thousand different actual structures. In terms of cellular proteins, that's true. There are a lot of viral proteins that look totally different. So that maybe should have been in quotation marks, the relatively few number of structures which are available here. So talked about domains, talked about motifs. If you have one of these particular domains, and you have figured out what the activity of a particular structure, now this domain has, now what you'd really like to do is use that information to understand what other things are doing. Usually that first information has come from genetics or biochemistry, as we talked about last time. But in this day and age, we've got all this sequence data. So how can you predict, given a sequence, what activity you're actually going to have? As I said, just from the amino acid sequence, we can't predict structures. But if you have a similar amino acid sequence for which there is a structure and a function, it's actually really pretty easy to predict what that is actually going to have in terms of structure and in terms of function. The rule of thumb here is that anytime you've got more than 30% of your amino acids just lining them up next to each other, that are the same, you're going to have the same structure and a very similar function. You can definitely have lower sequence similarity and still have an extremely similar structure. That's actually the example down here at the bottom. It's only about 19% amino acid identity, but the overall structure is basically identical, and not surprisingly, structure identical has a very, very similar function. Here, these two sequences are over 30% identical to each other. Both of these are proteases. They have slightly different amino acids that they cut. Yes?
I have a question. So, when you're looking at domains,
Mm-hmm.
Is an entire tertiary structure considered a domain, or is a domain within one tertiary structure? Are there multiple domains within a tertiary structure?
Okay, so I guess the question here is, given a tertiary structure, do you have one domain or multiple domains in an individual tertiary structure? The answer is yes and no. So it was just potentially why it's confusing. So you can have some proteins. That tertiary structure of that protein can be a single domain. You can also have a protein, again, one stretch from amino acid to amino terminus to carboxy terminus, which have multiple domains in it. So that will have a tertiary structure with multiple domains in it. You can also have a protein that says a single domain in it. Yeah?
So the different domains that you had in the previous slide
Mm-hmm.
for that tertiary structure, is it almost like subunits that have specific functions within that tertiary structure? Is that kind of how it can be thought of?
Yeah, so again, just to sort of paraphrase your question here, is if you have a multi-domain protein, can that have multiple different activities because you've got multiple different structures, which are there?
Like a subunit on a ribosome, you've got the large and the small, but this is a different thing. But I'm just trying to wrap my head around everything.
Yeah, no, I think it's a very, very important question here. And so with multiple domains, you are going to have multiple functions. If you say have one protein, like we were talking about before, one protein with multiple domains, that will have multiple functions. Some of those functions might be binding to DNA or hydrolyzing ATP or something like that. And we'll look at a lot of these proteins a little bit later on. But the way that those things work together, sometimes you'll have multiple proteins that are coming together and also are going to have that same kind of thing. That's going to be quaternary structure, however, and each of those domains will have a particular activity. So you can have some of both. Yeah?
So domains then like a very specific sequence of amino acids, and they can be, long or short?
Okay, so the question here is a domain a specific sequence of amino acids? And can it be really long or can it be really short? The problem is we can't predict what that stretch of amino acids is actually going to be, whether it's going to form a domain or not. Domains are always going to be in a linear piece of your amino acids, so from amino acid 53 to 124. But the important thing is that folds into an individual three-dimensional structure, which is then separable for all the other three-dimensional structures which are there. You can have domains as long as it forms a structure of as little as, I think the smallest ones are about 20 amino acids. Usually they're on the order of 50 to 150 amino acids. Does that make sense to answer your question? More or less? Okay. Good. But yeah, again, getting back to the question here about the individual domains, this is a domain up here. Oops, excuse me. Here's a domain. This is a domain here of elastase. It's a domain of chymotrypsin here. This is a domain. It's actually a DNA binding domain of two different proteins which are binding to DNA. Now, why do we care a little bit about that amino acid sequence similarity? We talked about protein families right back at the beginning. Those protein families, again, have been generated through either speciation, which is giving us paralogs or orthologs. Orthologs through speciation and paralogs by gene duplication in one organism, generating these large gene families. Those are going to have similar amino acid sequences, and then are also going to have these similar kinds of functions. Particularly if you know what they are, you can figure out what's going on there. So I wanted to give you a bit of a sense of scale. One of the things that I always have trouble with is sort of explaining how big a protein is, how small a protein is, how big DNA is, how small DNA is. And most of the time, when people see images like this in textbooks or wherever they are, it's not a really good to scale process. And so I love this figure because it actually does give you scale. And one of the things that it shows is you can have proteins that are pretty small and proteins that are pretty darn big. All of these are proteins. This particular one is a single domain, single polypeptide. This one, glutamine synthetase, I think is 12, if I remember correctly. But all of these, relative to DNA and RNA, they're about the same scale. Proteins have a tendency to be considerably bigger than that. And then here's a lipid bilayer, just to give you a bit of an idea what most membranes look like size-wise. So most proteins, about the same size as a membrane or bigger, considerably larger than the DNA, the individual nucleic acids. So finish up talking about a couple of examples of these proteins. I already talked about multimers before, dimers, tetramers in this particular case. This is a dimer that we will talk about later on. This is getting back to your question about, somebody's question about beta strands versus sheets. Here are two beta strands that are interacting with each other, two different polypeptides, again, starting with an N-terminus and a C-terminus, but interacting through hydrogen bonds between those backbone amino acids. That would be a beta sheet in between those two. Those two strands are going to form a beta sheet. So this is a bacterial DNA, actually a bacterial virus DNA binding protein. Here is a tetramer, 4 copies of a protein. This particular one is also from a virus. Anybody know what virus? Everyone got their shots this year? Flu? Heard that it's a really bad year for flu. Still get vaccinated. 30% protection, still a hell of a lot better than no protection whatsoever. I've been posting about this on Facebook and Twitter too. This is one of the proteins on the outside of the flu virus, neuraminidase. Has enzymatic activity. True for a lot of these enzymes and multimers. Turns out that the active site where actually having any kind of activity is between the individual monomers. And so that's what's shown over here. But 4 copies of exactly the same protein. You can also have multiple proteins coming together in one big complex of lots of proteins. The ribosome is one example of this. Another example is the DNA-dependent RNA polymerase. This has actually 12 different subunits that interact with each other to give you one big protein that's job is to take DNA and make RNA copies from it. And we'll look at this in more details a little bit later on. So you can have multimers, which are what we call homomultimers, same protein, multiple copies of it, or you can have different proteins all coming together into one particular structure. And to finish up, we'll now talk about the last of these, the non-structured proteins. Uh-oh, all these neat structures. How the heck do you have non-structured proteins? Well, strangely enough, and this is something that's been found literally just in the last couple of years, a lot of proteins that are in our cells actually don't have a defined structure most of the time. Or if you're doing biochemistry, you purify that protein and look at what that structure is. It turns out that it actually doesn't have much in the way of structure to it. It only has structure when it's interacting with another protein or a different amino acid or some kind of side chain as well. So a lot of times you see these pretty pictures, and I'll show all these wonderful pretty pictures. You'll see a particular structure. Great, that's wonderful. It's neat. It looks exactly like what's going on. Well, it turns out that things can have different structures. They'll have structures only under certain conditions. If they only have structure under certain conditions, that means they're only going to have functions under certain conditions as well, which makes this whole business really a lot more complicated. Isn't that wonderful? A lot more complicated than we ever thought was the case. And we'll talk about amyloids next time, and then some of the different individual proteins. Again, no lecture on Monday. See you on Wednesday. Your clickers will then start counting.